So thank you all for coming here Saturday morning, right? I know a lot of other things people can do Saturday morning, but I think this is a really important topic. And um, finally, we're starting to, in our lab, the work we're starting to do is really starting to pick up some attention. I'm going to basically talk about the research I've been doing for the last little more than a decade at the University of Miami. I came to the University of Miami, like I say, from Southern California. I was originally looking at air pollution research, and I got involved in algal toxins, which it just fascinated me. And I'm going to go over that today. So I have to say this: I don't have any financial disclosures. Everything I do is purely research. We have funding agencies. A lot of this work is funded by the Herbert Hoover Foundation. But otherwise, I don't work for a company. I work for the University of Miami. And, my goal is to do research to inform citizens of Florida. That's it, right? Okay. Thank you. Thank you. Thank you. Thank you. Thank you. Um, so today I want to talk about harmful algal blooms. Now, everyone knows what harmful algal blooms are, but we'll talk a little bit about it. And I want to particularly talk about cyanotoxin exposures. And then look at what we're doing in the lab. So we're trying to model these exposures, how they affect the brain, and then talk about some dementia risks to these toxins. So we know dementia is really a broad term that can mean a lot of things, but particularly I want to focus on Alzheimer's disease today. So harmful algal blooms are very diverse, right? So they're created by a lot of different algal species. We hear these terms red tide, green tide, or even brown tide, and these are basically created by many different types of algal species. And what we're finding is, depending on what species are dominant in these blooms, they create toxins, right? Certain toxins or different toxins. So, like I said, we heard of these things. These are just really kind of, internet pictures. These aren't my pictures, but they're really striking pictures on the internet showing how visually drastic these blooms are. And I think we see them every day. We kind of blow by them and say, "Oh yeah, the water's a little green, it's a little brown." We may stop when the water's red, but basically these things happen. And the thing is, we experience a lot of them in Florida, right? And they can vary in size, they can vary in how long they last, and that's where the issue is. So we talked about a recent really big algal bloom, and there's some really historical blooms that have been, in Florida that have almost been forgotten. There's the Blackwater event, 18-mile bloom. So these things, we're looking at these things in the lab. We want to kind of put together a broad historical picture. But for today's talk, we're really going to focus in on just cyanobacteria and what they produce that's harmful to us. So cyanobacteria, they're old, right? So they're billions of years old. So we can really truly call these our ancestors, right? They're the primary producers on our planet. So they're important for developing our ecosystems, right? So in certain conditions, like nutrient pollution, sewage, these cyanobacteria, which are naturally in the environment, can become, how we say, unchecked and develop these massive blooms, right? And this is not just a concern in Florida. This is a global concern. I think Florida, in my opinion, is really in tune with this because maybe we see them more. They're more frequent. I won't say that with a statistical number, but I think we're more in tune with them. But they are a global concern. And I love to show these two pictures. So this is the impact of these blooms, right? So you have some place like Miami Beach in Panel A, right, where you have luxury condos, you have tourists, you have homes, and you can see this in this major inlet, this major algal bloom, right? But in the bigger sense, in panel B, right, this is, I think, we can call like a kind of trans-state or international bloom. You have Lake Erie. So Lake Erie has a lot of nutrient pollution, and so you can see that this bloom affects not only Michigan, which is where D is, and that panel is for the city of Detroit. T, the blue dot, is for city of Toledo, and then W is the city of Windsor in Canada. So this is a massive bloom affecting millions of people, right? And for some reason, these things, they come, they go, and we kind of forget about them once they clear up, but they leave a long-standing effect. And what does that look like? So, we hear about the fish kills. It really affects our environment. So what these microorganisms do is when they become so dense, they really deplete the nutrients from the water. They produce the toxins. They can kill wildlife. Here's a picture of just one fish, but really these fish kills could be millions of fish that wash up on the shore. They impact our residents, our tourism, our economy. You can see these things, behind residents' homes. People who are fishermen or boat for recreations. They have to deal with these blooms, and for some people, this is not an option for them not to go out and have to work, right? So they have to go out year round. They have to deal with these things. So it's a constant exposure. And even though that they're not, dipping their hand in the water or drinking it, they're still exposed to it because it's aerosolized. So we'll talk a little bit more about that a little later on. But these impacts basically cost hundreds of millions of dollars. So they're probably equivalent to like a major hurricane. And states, around the country, they're investing money to mitigate these blooms by many different strategies. I won't go into them there, but I do want to show some numbers. So this is a research database. These probably aren't really accurate numbers because they're probably a little higher, but this is what the researchers found. And as you can see, they said Florida spends about 20 million. Ohio, the slide I showed previously before, they spend close to $100 million to mitigate these blooms. And this is huge, right? So, but what is the effect on us every day? So you and I sitting here. So really, you can go now and you can look at medical records and see the cost when people go to the emergency room. And they predicted that, an ER visit from exposure to these toxins, either through digestion or inhalation, can cause anywhere from $100 to about $15,000 for severe cases. So, this is not also a big impact on the economy, but also the health system. So this is where I come in. So this is very interesting to me. So I say, okay, how does this look like, And we started looking at cyanobacteria. Here's some warning signs that you can see around about blue-green algae. I was asked earlier about how can we be informed. There's a Florida algal bloom tracker. You can actually see active blooms going on. You see those are a lot of dots there in panel B. So it's probably not 100% accurate, but probably pretty good to warn a lot of people. And these things you have to be careful because they affect our whole body. Today I'm going to talk about the brain, but also our pets. Remember, animals, they are exposed to these things, and because their weight is a lot smaller, they're really affected much more, so it can potentially kill them. And so also research. So a big thing is Governor DeSantis gave about $14 million to start innovative technologies to try to help mitigate these algal blooms. So this is all a part of that money being spent. So this is a really serious problem. So everyone in this room pretty much understands this, right? So this is something that it's going to take a kind of a village approach to help stop. And our goal here is to kind of start at the research level to understand, how is it affecting our health, right? So I'm always asked, I don't live in Florida. I don't live by a lake. I'm not affected by algae. Yes, you are. So this is a warning sign from the Department of Health for the state of Nevada. So I can think of some drier places, but that's a pretty dry place. So if they're saying you have to be careful, so it's, and these things are global problems. So I really appreciate, like I said, I moved here about 13 years ago, and I think Florida, in my opinion, is probably really leading the charge on this. A lot of the research, the cutting edge research, is coming out of Florida about these topics. So when we're exposed to algal toxins or cyanotoxins, these are probably mostly due to frequent blooms. What happens is you have some kind of nausea, vomiting. You can have short-term memory loss. So it affects the brain, it affects the body. Rare cases, you can have organ failure, but these are really extreme cases, right? Here's just a diagram. This is a publication in 2019 of just the distribution of different types of algal toxins produced by cyanobacterial toxins and where they're located. So you can see Florida at the bottom, California. So coastal areas with a lot of water, you have this high reporting of particularly microcystins, which is really popular or really well studied and really, really common cyanobacterial toxin. And it's also a neurotoxin, and it affects our liver. But today, this is some, I won't go into all the chemistry here. This is just, these toxins are different. That's all I want to say on this slide. But I want to say what we study in our lab is completely different. So these toxins that are on the left-hand side, the microcystins, anatoxins, they have really cute effects. So if you're exposed to them, you know right away. And you get a lot, you really know. I want to talk about non-protein amino acid toxins. These are parts of amino acids. It's just really a building block of protein. These things are released into the environment. They're called non-protein because they don't really exist in protein itself, but they have the structure of an amino acid. And the thing is, what we're showing, I'll show you a little later, is that when you're exposed to these things, they hang around in your body for a long time. And that's what we think they're able to get into protein and cause some really acute effects. Really chronic toxicity. So today I'm going to talk about a well-known neurodegenerative agent called beta-N-methylamino L-alanine, which is BMAA. And kind of its origins and what's its significance in Florida. And I also talk about the next molecule down, which is very similar in structure, but has a completely different mechanism, but you end up at the same place. And when you're talking in toxicity. So how are we exposed to these cyanotoxins? Well, mainly we believe we eat them, right? So we believe that, they get into our food web, they can bioaccumulate, through the marine food web, and we ingest them. We have shown this in the lab. I'll show a little bit more that it is true. It does these toxins do bioaccumulate. They go all the way up to apex predators, so sharks, dolphins, and, people who consume these large fish will get a lot of them. And we believe it's a risk factor for Alzheimer's and ALS. So how does that look like? So I told you about primary producers, the cyanobacteria. So basically what happens is when these toxins are created, they basically accumulate up the food chain, right? So one thing eats another thing, another thing eats another thing, and you end up, with really high amounts in these apex predators, right? So we've tested sharks, we looked at brains of dolphins. We'll talk about that a little bit. So we actually looked at shark fin, and interesting, the data, I don't show this data here, but we also looked at shark fin supplements, and it's also in the supplements. So it's something that's there, it hangs around. So this BMA can go up the food chain, reach these apex predators, and deposit in their tissue. And ultimately it gets into us because we eat shark, we eat these things in the food web. There's blue crab there. Blue crab is very delicious. So we get them too, and so now when I talked about the university and the society, in general being more open to this research now, is because they're now seeing that there's a huge link between marine health and human health. So now they're trying to synergize the two to understand, which makes a lot of sense, because without really great water quality, you can't have good health. So Joe talked about Adam. I won't go into detail because he's an expert here, but I will say that we saw this technology, and we were so impressed by the press release that came out with the concentrations that they were detecting cyanobacterial toxins in the air. It was amazing. So, yeah, I mean, there's a lot of different hypotheses how these toxins get into the air. Me as a pathologist, I don't completely understand them, but basically, it has a lot to do with the physics and chemistry of the water and how they're dispersed. But they're truly in our air, right? And you can see this by apparatuses like this, and even there's studies saying that it's on home AC filters. So if you take your AC filter, you can actually extract these things from these cyanobacteria, these toxins from your filter, which I think is a great thing, because if your filter is stopping it, then that's great in your house. But so we wanted to say, okay, this stuff is in the air. Can it get into the brain? So what we did is we designed an experiment to look at cyanobacterial toxin exposure through the nasal epithelium, and this is called the olfactory pathway. So when you smell a molecule in your nasal epithelial, there's little nerve endings that go down that kind of register these molecules, and they send signals back to the brain. For toxins, this is pretty much like leaving the gate open, because one, there's no blood-brain barrier there to stop anything from getting in. So it's a really a very vulnerable part of how our brain has developed to have this really complex olfaction. So we tested that, and what we found, so we took residents of Florida who had Alzheimer's disease. We started there, and we took one control, and basically this is how those nerve endings look and be. So this is a brain of a individual that donated their brain to the Miami Brain Endowment Bank, and we were able to collect these really precious specimens. And B, you can see the numbers 1 and 2 in panel B are where those nerve outlets would be, where the toxin would interact with. So we collected those, and we ran them through a really sensitive assay to detect these toxins. And what we found is that not only are these toxins in those parts of the brain tissue, they were relatively high, and they were also in all cases. So they were in people with Alzheimer's and people without Alzheimer's, which makes sense, right? Because why would someone with Alzheimer's have more of an exposure than the normal person, right? Right? Because the normal persons, they're out, they're working, they're boating, they're fishing. But the key we have to look at is when we look at the structure of the brain. the individuals with Alzheimer's disease that had a high amount of toxin in their brain, if you look over at case 5 and 2, the tissue was almost degenerated, right? So not only were they suffering from Alzheimer's disease, but they're also suffering from this neurological or neurotoxicity from these particles. So we published this in Toxicology Reports as a case study, and we did get a little media attention about this, but it kind of went under the radar again. And so we thought, okay, we really need to just, kind of push it more. I thought this was really striking data, but again, it was only 5 cases, right? So we need to do more. But as you can see from in panels D, E, and F, that tissue, you see the white parts is basically, the white represents that there's neuron loss, so there's holes in the tissue. So the more intact tissue would be more pink. So we said, okay, let's do this. Okay, we need to start thinking about preclinical models to model these toxins, right? So we know they're in the environment, we know they're in the air, we know they're in our food chain, we know we're being exposed to them. We know they're toxic because of their chemical structure. We put them on cells, they die. How do we convince the scientific community and abroad that these things are actually causing disease? So I talked about BMAA again. I won't go through this a lot, but I will tell you that the molecule is very complicated because it has a lot of different ways it can cause toxicity. And the way what we're gonna focus on today is mostly that it can incorporate into protein and cause proteins to tangle up. So you have a normal protein, this BMA can insert itself in there unnaturally and cause a normal protein in your body to misfold itself. And once that happens, that protein kind of lumps together with other proteins that are similar, and it causes what we call an inclusion. And those are bad for the brain because you can't remove them. They become inflammatory, and then they lead to neighboring cells pretty much dying. So we're going to talk about that because we consider this molecule as a neurodegenerative agent, and I'll explain a little bit more about that as we go along. So I talked about protein incorporation, right? So basically, experiments have shown that if you take the toxin, BMAA, and you insert it in a specific residue in a protein, that it'll misfold. And what they've done in people with Alzheimer's disease, for people with Alzheimer's disease, AMALA, is they've done this in vitro. So if you put this BMA molecule in an Alzheimer's disease protein, that's really toxic to our brain already. It becomes more toxic. On the other hand, we all know of ALS, right? So SOD1 is superoxide dismutase 1, which is a very important, basically antioxidant for our body. So if you put BMA into that, it's a protective protein. It loses its function. So this is just saying, like, these chemicals, these toxins are very, very intricate in the way that they make us sick, right? So there's a lot of different pathways that we can follow. So we have to focus on a few pathways that we think will have the greatest reward in proving our hypothesis that it causes us to become ill. And so what we did first in the lab is we started really basic. So we took mice, we injected them with BMA, and we waited, we watched to see if it goes to the brain. And panel A is a radiograph. This is basically a brain slice that has been exposed to film, like a film exposure. And all those black dots are the BMA, right? So we put a carbon label tag on the BMA, we injected it into the mice, and we waited. And we saw over time that the amount of BMA that was injected in entered into the brain, and it actually bound to protein. So it bound to protein. So what does that mean? So you don't want a toxin to hang. The really take home of toxins is that they hang around, right? Anything in your body that hangs around for a long time is not good. So BMA is able to go into the body, go into the brain, bind to protein, and hang around. So that is, that shows us that this molecule can create a reservoir in our brain. So we talk about multiple exposures to harmful algal blooms. So if you're constantly being exposed, let's say through an aerosol exposure, these molecules can potentially build up in your brain, and ultimately you have this effect, this long-term effect, which we're still trying to figure out, but we have a pretty good idea what it does in the long run. So the next step was to see if we could really reproduce disease. So we went to a really higher model. We went to a primate model, and this was done by giving a primate algal toxins and 2 doses, basically a very high dose, which is equivalent to a dose that was received by a Chamorro villager in Guam. And what I mean by that is that I'll talk a little bit more about that later in the talk, but Chamorro villagers actually were exposed to a lot of cyanobacterial toxins and developed a really special type of neurodegenerative disease called ALS, Parkinson's dementia complex. So we tried to model that in this primate. And, we were able to reproduce Alzheimer pathology. So the top panel B is the vervet, and those are just immunostains for amyloid. This is really the key protein in your brain that they use to diagnose Alzheimer's disease. So one of the 2 keys. So on the bottom is human. So this is a human with Alzheimer's disease, and on top is a vervet. So it's very similar. So if you give them this toxin, you can reproduce these proteins in their brain. And we think it's because, as we said, the BMA is going in, it's binding to these proteins and causing them to misfold. This is another sample of slice of brain. This is a large section showing early Alzheimer's disease pathology. I won't go too deep in the anatomy here, but if you look at the far end with the arrows, and we saw the BMA growing in the tissue. So this is a stain for a protein called tau. So in Alzheimer's disease, you need 2 types of protein to be abnormal in your brain to secure the diagnosis. You need amyloid beta, which we now have drugs available to treat, and hyperphosphorylated tau. We don't have any drugs available to treat this. And you can see these tau building up in the areas of the brain. This area of the brain is actually the amygdala and entorhinal cortex, which are the first regions of the brain affected in Alzheimer's disease. The amygdala, if you know someone with Alzheimer's disease, you may know they have, really outbursts of emotions, right? So this would be that pathology here. So amygdala kind of controls our emotional centers, like and aggression too. And this is just a higher magnification of those lesions in that area of the brain. So they're bona fide tangles in the neuron cells. They're really staining really dark black. These are antibodies that identify only that protein. And basically, from top to bottom in panel A, you see a normal accumulation of this pathological protein until the neuron dies completely. Now, the good news is, always the good news. So remember I told you that the BMA can go into these specific protein residues and cause the protein to misfold. So what we did is we gave L-serine, which is an amino acid, at the same time with BMA, and we're able to block this. So almost 50, this is 20 to 50% block. So it shows that these toxins can go into the protein and cause the the protein to misfold, but it also can be blocked, right, by a, um, a simple amino acid, L-serine. This L-serine is now in clinical trials for treatment of mild cognitive impairment and things like Alzheimer's disease. So when we talk about dementia risk, so we talked about, we looked at some in vitro models, some case studies. We looked at some animal models, but what does that mean for you and me, right? So, we're not mice, so we're not in a controlled environment. Well, at least I hope we're not in a controlled environment, but we're not in a controlled environment. So I talked about Guam, and I really want to highlight this paper because to me it's just a landmark paper. This was published in Neurology. This is by Paul Allen Cox, who's a director of the Brain Chemistry Institute, and also Alifer Sachs. So I don't know if you know Alifer Sachs. He wrote the book, The Man Must Baked His Wife for a Hat, right? I hope I pronounced that right, but I get a little nervous. So they basically found that these cyat neurotoxins, which were cyanobacterial toxins, BMA, were biomagnifying in the food chain there. And the local villagers were eating a lot of it by consuming these flying foxes, right? So as part of a cultural practice, they would hunt foxes who ate these cyats and got a really large dose of BMAA, a cyanobacterial toxin, and they would develop this really strange disease, which is like basically a mixture of many different neurodegenerative diseases we know in the clinics today called ALS, PDC. And basically these individuals have dementia, they have Parkinson's disease, they have Alzheimer's disease, they have progressive supranuclear palsy, all wrapped in one box. So, and this is from eating this toxin. And the reason why this is so groundbreaking is so many physicians and pathologists went there with the wrong assumption that, well, I won't say wrong, they had a good assumption, but what happened is they looked at everything except the environment. This is how powerful the environment is, what we do, what we eat, how we live our lives, what we consume. And what's interesting about this is once the flying foxes were almost extinct, the disease dropped. So they weren't eating them anymore. So they weren't getting exposed anymore. And how does that look? So we talk about that exposure. We talk about them eating the cyat tree. So basically that BMA magnified up, multiple folds into these villagers, and they developed these plaques and tangles that I showed you previously that we were able to reproduce in the vervet model and the primate model. And so it was, this is really striking. This is a real documented human exposure on an island where they have 50 times the incidence of ALS because of this. So, when people say, oh, well, these things won't make you sick, you can just quote this study, or this multiple studies. I mean, this is, a real disease that, has an IDC-10 code where it can be diagnosed, and it's in textbooks. So it's really interesting. When we look at humans outside of Florida, outside of Guam, we look at humans in Florida, and we looked at, this was a research done by Dr. Deborah Mash, looking at a larger set of individuals with Alzheimer's and ALS. They were able to find comparable amounts of BMA in the brains of these individuals with these chronic diseases. Interesting enough, Huntington's disease, which is caused by a gene mutation, they really didn't find a lot of the of the cyanobacterial toxins, right? But for the individuals with these sporadic neurodegenerative diseases, they found a lot of the toxin in their brain. They also tested cases from Canada. It was there too. So this really stimulated a lot of hypotheses about maybe these things can really cause Alzheimer's disease. And I want to show this. I think this is really important. This is really recent data. So this is prevalence of Alzheimer's disease, and this is the 220 map. But Miami-Dade County was number one. It's consistently number one, right? So just right across the street, right? And there's a lot of things that go into looking at calculating these numbers. So I do tell people, it's not really a sheer number, but there's a lot of different calculations that go into this. But what's interesting is if you look at 2020's map and that cyanobacterial toxins exposure map that I showed you, those points in those of Alzheimer's disease and cyanobacterial exposures, they start to look a little similar, right? So this is just by eye. We really have to go in and do a really quick and, really in-depth analysis to really figure out, okay, but I can tell you, California, Texas, southern states that have a lot of coastal exposure, you can see, and even in the Northeast, Massachusetts, New York, there's where the exposures are reported a lot, you see a lot of Alzheimer's disease too. So there seems to be some epidemiological trends here. Obviously I'm comparing two pictures. It's not a statistical analysis, but it's really striking to look at them. And I'm sure people can find correlations in that data set. So what we did is, we measured this. I showed you some of that data before. We measured it in healthy controls and people with Alzheimer's disease. So the brain up front is an individual with Alzheimer's disease. So I talked about looking at the tissue with those spaces, those white spaces in the brain as a whole. So basically, Alzheimer's disease, neurodegenerative disease, the brain basically shrinks. You lose density in the brain. And depending on where the density is, you have a really specific neurological effect. Memory is lost, executive function. So we actually, like I said, we tested these compounds in brains of Alzheimer's disease, and they weren't they were very low in control patients. And so we used that study to say, okay, well, people say, well, you pick 12 cases, maybe those cases were picked at, they're there, but, you always get pushback. So we went on a ledge and we did something really, I think really kind of maybe left field, but I think it was really, brilliant that using this marine sentinel species, which was kind of adapted into the lab when I got there. And what we did is we did the same thing in dolphins. We said, hey, dolphins, they live in these blooms, right? So if anyone's going to be exposed to them through the food chain and through their lifestyle, it would be a dolphin. So we took dolphin brains. We had a very nice collaboration with Woods Hole in Massachusetts. We were able to get some samples from Cape Cod Bay. So Cape Cod, and we compared them to Florida. And Florida had a lot more algal toxins than Cape Cod, but they were still there in Cape Cod Bay too. We had about 3 times more here. But I wouldn't preface that as being us being worse or less. The point is that the exposures, even in Cape Cod Bay, it's still there. And we looked at the neuropathology of these dolphins. So, what we try to do is do a comparative study. We try to compare humans to dolphins. This is really not too difficult to do because dolphins are also mammals. The brains are relatively similar, but they're different enough to be challenging. So we tried to just map what would happen in a normal Alzheimer's disease brain to what was going on in the dolphin's brain. And we see that the dolphin has similar, and these are some representative images of those tau and amyloid beta plaques I was telling you about. They have a similar distribution of these things in their brain. And we published that in Toxins. It got some really good attention, but again, it just kind of, it was a flicker of light. So people said, well, they're dolphins, and it really wasn't a broad enough picture. So, as always in research, we keep pushing things. So we had to do things bigger. So instead of looking at histological sections, small sections of brains, we took the whole dolphin brain and we just sequenced it. All the gene expression going on in that dolphin's brain at one time. And we searched for signatures that were related to Alzheimer's disease. And this one got a lot of attention. So this one we published in Communications Biology recently, a couple months ago. And basically what we did is we looked at a really specific population of dolphins in the Indian River Lagoon. We tested for BMAA and another isomer that I talked about called 2,4-DAB, which is similar to BMA, but it's a structural isomer, so acts a little differently, but it's still a neurodegenerative agent. Interesting enough, we found that, even in the Indian River Lagoon, there's different areas that exposure is different for these dolphins. So we did this over a decade sample. So the research didn't take a decade, but the sample was over a decade. So we grabbed, we had our collaborators at Hub's Research Institute, Hub SeaWorld Research Institute, carefully select 20 dolphins over a decade that they felt that the cause of death was really not too clear. They may have had some neurological symptoms when they passed, right? This is all more, not really the necropsy reports, but since they were on site, they were able to see these things. And we did this analysis with them, and we found that, if you take these dolphins and you divide them into two groups, those who were found stranded or beached outside of bloom season and those found inside of bloom season, the toxin exposure is much, much more in bloom season in their brain, which makes sense, right? Makes perfect sense. So, this bar graph really doesn't do it justice, but if you look at the median or middle value, it's about 2,900 times more, the exposure in bloom season. Inside brain tissue. This is not liver. This is not any other organ. This is actually in the brain, and we know maybe only 1% gets to the brain for these things. So really a large amount in the brain. Like I said, this is a really busy graph, but I show this to demonstrate, and on the left panel, you see all those square tiles. These are the actual genes that are being expressed. So, and this is only a subset. So we only took about 900 or 800 of these genes, right? And those genes that are green, those green tiles, they're increased in expression, and those ones that are orange, they're decreased in expression. So if you look at NB, which is non-bloom season and bloom, you see a totally different profile. This is going on in their brain. Totally different gene expression profile, right? Now, we know seasonal change has effects on our body. We all know about changes from summer to spring, which is very important for our biology. But when you add in a toxin to this, it becomes really complicated and probably not good for the body. So we are able to identify some really target genes. Genes are basically coding regions that produce proteins. So we talk about proteins misfolding. So these would be the upstream biological components of the protein. So and basically you have ideally a different protein profile going on in the brain during bloom season. And we looked at that. So we looked at the major risk factor for Alzheimer's disease, which is ApoE, right? So bloom season, it's maybe 2 or 3-fold higher, right? So if you can think about this, if you have a if you're predisposed to develop a condition and you have a particular gene that makes you more susceptible, then if you're exposed to a bloom season, this would magnify that. We looked at some other markers. These two are, I put them there to show that this is really happening across time, right? So these are cross-sectional studies, meaning at one time point. But when you add them together, the dolphins that were found in 2010 versus the ones in 2020, the ones in 2020, they're getting worse, right? So if you look at the parallel lines, and then the ones in the bloom season here in the gray, they're not changing. But when you're exposed to these toxins repeatedly, bloom season after, it's priming you to have a more increased risk or increasing your risk factors. Sorry, I think I'm getting close to it. So, yeah, these are major Alzheimer's disease protein markers. The blue and green, the dark blue and green are the bloom season with the toxin. They're increased for tau, and there's another marker here that we looked at. That's this is called. It's basically the protein's called TDP43. The significance of this protein, I told you, in Alzheimer's disease you have amyloid and tau, but if you have this third protein, your disease is twice as fast. So you can see that these blooms are, at least in the dolphin. So remember, these are dolphins, and we're comparing them to a human condition. But ideally, and theoretically, yeah, exposure to these things is accelerating pathology that's linked to these disease. And we looked at the tissue again. And what's interesting about this, I won't go into too much detail, but now there's a clinical marker for Alzheimer's disease. It's a phosphorylated tau at a phosphatidylserine residue called 217. So this is mean the tau protein has a modification on this specific residue, and the clinicians use an antibody to measure this, right? And if you have this type of phosphorylation, it's correlated with disease in the clinic, and you can see it in the dolphin here in panels F, G, and C. So this was striking. So we went ahead, like I said, we put this together. The editors at Communication Biology were really great about this paper. It got a lot of attention. I think, for a journal article, I think it's more, right now they stopped doing the metrics, but within a couple months you get about 20,000 views. And that usually doesn't happen in science, not too often. So it was ranked really high. People really received this data really well. So, and the reason why I say this is not to promote the paper, but to promote the general thought of the scientific community that these type of studies are very important, because they're looking at anything to figure out what's causing these neurodegenerative disease. It's really a ticking time bomb, and I think right now environment's winning. So environmental exposures, because we've done a lot of genetic work, and we're just not finding clear links. We feel genetics is a big part of this, but we really think the environmental component is just as equal, if not more important, in accelerating these diseases. And cyanobacterial toxins may not be the cause of these diseases, but we feel like it may be one cause, and it's just definitely not helpful. So I know I used a lot of terms here, and I do apologize. I mean, this stuff is like a different language, but I want to sum it up. And I think this is a really good photograph to kind of show you visually what's happening in the brain of an Alzheimer's disease patient. This is taken from the National Institute of the Health of Aging. This is one of their graphics. So basically in your neurons, those big things there, those big pyramidal-shaped things, this tau protein is misfolding. It's colored there blue. So it's misfolding in the protein, and the protein's misfolding, and it's causing the neuron to die, the process of die. On the same time, you have these amyloid plaques that are building up outside of your neurons in the periphery. So there's drugs now to remove this, but you can't remove tau from inside of a neuron, right? So you can potentially remove something that's outside of a neuron and clear it, but you can't remove something inside of a cell. And this is what we show these toxins to cause, and then the accumulation of this tau. And also, I talked about TDP-43, those yellow circles. If that's there, it's even worse. It accelerates it. So this is basically the mechanism of what's happening in the brain when you have Alzheimer's disease. And when you add cytotoxins like BMA, they increase the expression of tau, and they increase the expression of this TDP-43, which clinically we predict could make the disease worse. And yeah, just lastly, so in the dolphin, when you enter into bloom season, you're affected, you're hit with these toxins. It causes the gene expression in your brain to change. The synapses between your neurons, which neurons talk to each other with, they're dysfunctional. And basically, this is increasing the risk of Alzheimer's disease, or Alzheimer's disease-like changes in this animal. And I think with that, so I just want to summarize. I know I went through a lot, but it's literally a decade or more of research. But so just the take-home notes is these blooms are a major economical and public health problem. Florida is a epicenter of these blooms. There's a really high risk or potential risk of exposure in Florida. Harmful algal blooms produce toxins that could potentially cause neurological illnesses, or long-term neurological, chronic neurological. They cause neurological illnesses, but they could potentially cause these chronic neurological illnesses like Alzheimer's disease. And exposure to HABs are likely to increase the risk of the occurrence of these diseases. So with that, I want to thank, first of all, thank you all for being here, and my collaborators and mentors at University of Miami, my collaborators at the Brain Chemistry Labs, and who've done a lot of this work as well, and my collaborators at Hub's SeaWorld Research Institute, who hand-selected those dolphins and worked with us to get that paper out. And with that, I open up for any questions. Thank you. Okay, so Iowa has a similar condition. I want to say like Lake Okeechobee. They have a lot of agricultural work going on along the coastline, so they have a lot of nutrients going into the water that causes their blooms, and potentially sewage runoff. So two of the big stimulators of blooms are, nutrient pollutions, right? So, and then also the climate warming. Right, so increased water temperature. There's some other factors, but we believe it's mostly due to nutrient pollutions. So that's being nitrate and phosphorus going into the water, fertilizers.
Over here.
Oh, sorry.
Hello. Like a lot of items, like the FDA will approve chemicals in food that you obviously would have to eat an extreme amount, like for like Splenda and sucralose and such. They limit to, I think I looked up the other day, a 200-pound person would drink over 24 cans of Coke, a day. It would be an excessive amount beyond, which could cause issues like, potentially cancer. I fish in the Gulf, and I eat like 2 meals of fish a day, 5 ounces each meal. What levels are we talking about here of cyanotoxins that you would have to intake to actually pose the conditions that you're discussing here, I guess?
So the research that we did using the primate model, the vervet model, was really, it was a high dose that was consumed through Chamorro villager, and this was a chronic dose that happened over like 140 days. We don't really know. In that model, we use. I'm thinking it's. I don't want to quote the exact number, be wrong, but it's a very high amount, right? But what I will say one thing. We don't know exactly what dose will lead to someone having full-blown Alzheimer's disease, but we do know that these toxins, they don't act alone. They act in synergy with other toxins like methylmercury and other algal toxins that can synergize them. So it's very difficult to give an exact number because we just don't have human studies to dose. I mean, I can give you the amount for a primate or a dolphin, but it's too hard for humans to say.
And obviously I wouldn't be able to calculate that. I mean, but what I have read and heard is that the cyanotoxins are mainly accumulate in the liver, and that in the organs of the fish, and that the flesh, the fillets of the fish, are actually been deemed safe. Do you have any thoughts on that?
Yes. Physiologically, that makes a lot of sense because the liver is going to clean out a lot of toxins. So most toxins are going to be accumulated there. But it also depends on what level of fish you're eating, how high up they are in the food chain, what they've eaten, and their geographical location. It begins to get complicated. But you're right. The distribution of toxin is a lot different. But I would say, for example, for sharks, people eat shark fin.
Yes.
And, that we found in really high concentrations.
And I have read that too, like the pelagic fish, the huge fish, like cobia, for example, grouper, large grouper.
Grouper, yeah.
I mean, they accumulate more mercury. And I just saw something that came across some of the fishing blogs that I'm on that said avoid cobia, period, because of the levels of mercury. Well, that's like one of my favorite fish.
Yeah. Yeah, the seafood's, it's good.
So, I guess I eat a lot of most of what I get are the smaller fish. So it would be more like the bait of the shark, the small snappers and the sheep's head and things like that, that probably don't have that level because there's the smaller fish, I guess. But
well, I can say, just to answer, like for cyanotoxins, I mean, you really want to, like, fresh water is probably going to be a little more risky than ocean caught food, right? So methylmercury is probably really a big concern, and we know how much the limits there. I think it's about 1 microgram per gram with that, but yeah, for BMAA and these algal toxins, I'm just not sure what's the recommended amount. I would say 0, but I mean, obviously they're there, right? And our bodies are really resilient, and the work that I do is kind of focusing on those small doses over time. What do they do? So,
Tests for like if you had a fillet of fish in front of you. I mean, I'm thinking of like this would come out with some kind of like a testing kit or something that you could actually identify.
So they have them. It's just getting it to the consumer, where you can test as much fish as possible. I mean, methylmercury they'll test for it because it's more regulated, but the BMA we have to really convince people to make it, a part of a daily routine but it should be. You're right, it should be.
I think you said that once the DNA for Alzheimer's gets in the brain, you can't do much about it right now. But then you talked about this L-serine that's in clinical trials right now that can protect the brain. Can you just explain briefly what hope comes from that potential medicine and how it would work?
So I'm not too familiar with the clinical trials, but I can speak about the Vervet model, and basically what it does for that particular toxin is that it blocks the incorporation of it into protein. So so basically you're looking at, L-serine is good for brain health in general, but when you're faced with something that is a specific mechanism, it could be a way to protect yourself from this accumulation. So, diets rich in, protein would help, or you can take it as a supplement too. That's because it's a normal part of our diet. We use it as experimental. Say, okay, if the toxin is going to go into this residue, can we compete with it with L-serine? And it seems to work. So.
First of all, Dr. Davis, thanks for taking your time and coming here and taking up your Saturday and coming over from Miami. My question is, there's a certain philosophy out there called One Health philosophy that you may be familiar with, where human health is connected to animal and plant health, which is also connected to the environment. We're not doing cutting edge research, which, boy, I wish I was. That's fascinating. But what can we do to help improve, just as the general population? Any recommendations on what we can do to help improve the One Health, or is it awareness? Is it any suggestions?
I think exactly what you're doing now. Yeah, awareness. Water keeping. I think for us, I mean, I look at medical effects of these things, right? But there's keeping people safe, testing. There's a lot of things we can do individually. It's difficult to say you can't remove cyanobacteria from our environment because we need them, but potentially, advocacy work, letting people know that this is important, and just kind of really getting the word out. I mean, you talked about ocean health, human health, environmental health, how they all go together, and I think, for what we do in the lab is more really hyper-focused, right? It's more hyper-focused on looking at a particular disease. But I think for general well-being, potentially, warning people. A lot of people don't know. They see algae, they swim in it. It looks healthy. It's green, right? So, just letting people know, spreading the word. Nutrient pollution is another thing that we can deal with, but that's a little more complicated. Monitoring, and test your water. You can buy water test kits. They may not have this particular molecule, but you can test water, and you can talk to your local government and say, hey, this is happening, and nicely, because they want to help too, right? They want you to be happy. So.
Welcome back, Dr. Davis. It's a pleasure to see you again. The last time we met a few years ago, Paul Cox was here, and he kind of burdened me with giving some medical education lectures to our healthcare system. I prepared a series of 3, and after they heard the first one, they uninvited me for the next 2. Apparently it was a little too scary. We have some good news, and now Parkinson's has become a reportable disease. But believe it or not, in the state of Florida, ALS is not a reportable disease. At the time we last met, we had 2 bulbar ALS cases in my small office in Naples. That's like finding a dozen hen's teeth walking down the street. And it's still not reportable. And I think one of the things maybe we should try to do is put a pressure on our government to start at least helping us with the epidemiology of these things. What do you suggest on that topic?
ALS. I'll speak to ALS and algal toxins. There's as I showed you with the Guam study. There's a really strong strong link between this toxin and ALS. I mean, ALS could be caused by a lot of different things, but as you say, having two bulbar onset cases, bulbar onset is a subtype of ALS in the same office is really type of, it probably would maybe lend to a shared exposure, right? So, yeah, it's difficult. I think, we just need more research. These diseases are sporadic, so they just happen. But they call them sporadic, but it's just really no clear way to convince people that unless you give them a toxin and you see it. But I think studies like that. I mean, I would recommend probably a case study of that to a journal of neurology. I think that would be a well way to let that out. So.
Doctor, I have a lot of students here from my biology classes,
Sure.
and
Sure.
they probably don't know what ALS is. Or
Sorry.
but I just get you to, kind of bring them up to snuff on what we're talking about.
Sorry. So yeah, sorry. So ALS it's normally called Lou Gehrig's disease, and what happens in ALS is the motor neurons in your brain degenerate, and it causes you to have a paralysis, and eventually, because of that degeneration, those motor neurons in your brain. You generally succumb to respiratory failure, right? Because the lungs can no longer move. We just saw a really recent famous actor who passed, Eric Dane, who had ALS. Really rapidly progressive disease. Impacts is very horrible. It hits people in the prime of their life, very sporadically. We're talking athletes, people who have really great health. It doesn't matter. It hits them. And then with 6 months, some of them even after 2 months after diagnosis, they pass. So it's really a horrible disease. It's not as common as Alzheimer's disease, but it's really traumatic on the family. So that's basically what that disease entails. And there's a famous baseball player, Lou Gehrig, who's kind of the face of it. You can look it up, and it explains a little more about it. But Stephen Hawkins also had a form of ALS. He had a different form, which allowed him to live a lot longer. So.
Well, thank you for your presentation. Okay. Thanks for your presentation. So, I understand eating fish that's contaminated might contaminate us. But what's your opinion about the blooms on the river? How close does one have to be in danger of exposure, maybe disease? I mean, it's in the air, right?
So proximity to active blooms, it's hard for me to touch. I think we'll touch on that a little later today, right, with the Adam report. But the case study that we did using the postmortem cases, these individuals were less than 100 meters from a fresh water body. But that doesn't mean they were exposed by that water body. So it's really hard to say. I think rule of thumb is if you can smell something, you're probably exposed to it, right? So if you can't smell it, are you exposed? That's a really good question. I think we need more research to figure that out, and I'm definitely not an expert to give that number, but I think closer proximity would be, like if you can smell it, then you're probably being exposed, right? Sorry.
Hey,
Hi.
I'm a student at FGCU, and one thing that our university does really well is research. But how would you suggest, students maybe looking to get into this kind of research? Because I'm taking microbiology right now, and this is something that comes up. But I interact with bio students, marine science students, even journalism students and stuff who might not be aware. And how would you suggest, students kind of get more involved in this and even become more aware of this? And what can they do, either research-wise, internship-wise, to do this kind of stuff?
So, with research, I think seek, mentorship within your university. And then what we always do in academia is we can collaborate. So if there's a particular lab you're interested or type of work, you can have whatever lab you're in or your mentor reach out to that lab. This is really unique research because it's based inside of a biorepository, which we have access to tissue, and we're able to design these experiments. But that doesn't mean you can't do the same thing. It just there's some regulations, but my main thing was reach out to your mentor or professor, and then have them kind of communicate with the lab you're interested in working with. That's generally how this discussion starts. So.
Thank you for coming today, Dr. Davis. Mike, I have a question, but I also have anecdotal evidence. I live in Hendry County, and they've been in disbelief about the amount of blooms that we're having, because I reported one and a couple of them. And the very first one was really, really bad, and I live a mile away from the Caloosahatchee out there. We had cyanobacteria in our pond. So that, just to give you an idea of how far I know it travels. Yeah. It could be farther. So the other is there a way to, do we have a way to measure this in humans, or just water and other mammals?
Water is very easy to measure these molecules in. Because the matrix is very uniform and simple. Once you move into complex tissues, it gets more difficult. And then there's the possibility of molecules that are could interfere or interact with the assay, could represent or inflate values. So it gets a little different. But if you're referring to an individual testing themselves, theoretically, you can just test your own blood for any marker you want. But, would the insurance pay for that? Probably not, unless you report and you're, maybe sick under the care of a physician, and they say, okay, I want to make sure you aren't exposed to brevetoxin. So there are clinical ways to do this. They're just generally kind of specialized. Ideally, it would be great if everyone could test themselves for everything periodically, all types of toxins, right? But it could theoretically be done, and I know some instances it is done. For the molecule we're looking at, I don't think I have no knowledge of it being tested in the clinic, but I know for brevetoxins, microcystins, they will test. So. Thank you.